Hey guys, I thought I should show my face on here more often. I do realize I'm upside down and I don't know how to fix it. If you haven't noticed, I'm not very technologically advanced. So yeah, hi. We're gonna do Meiosis today, chapter 19, and I'm gonna go ahead and flip this off of me down to our notes here. Okay, before we get started, I want to do a little quick review. I have some pictures here just with some terminology. So a single chromosome, like you see here, we call that a chromatid. And then once it's replicated and you have two copies of that chromatid, we have sister chromatids and you've got the centromere here in the center. And then, we're going to hear this word a lot today, homologous chromosomes. You get one from mom and one from dad. So these are a pair of homologous chromosomes here. So I'm not going to go through any of the pre-lecture or key concepts today. You guys can read through that. But I will say doing this ahead of time is going to be very helpful for today. And we will go ahead and start right here. So curse whoever named these, and I'm sure there's a reason behind it, but mitosis and meiosis sound very similar. So kind of a silly way to remember it is mitosis that we learned about in the last lecture. Just think about my toe. And so this is going to happen in parts of your body, like for example in your toes, to replicate cells, for healing, growth, all of those things. And then meiosis, you can think of cis, because when we talk about this one, we're talking about sex cells, gametes, eggs and sperm. So, meiosis. I don't know if that will help, but there we go. So a quick review of mitosis. You end up with two daughter cells that are identical. And meiosis, you're going to have some different results. So what meiosis is primarily used for, I think only used for, is sexual reproduction. And what you'll end up with are four unique or different daughter cells. So mitosis, you get two identical cells, and meiosis, you'll end up with four unique or different cells. And the daughter cells are going to be haploid, meaning they have half the amount of chromosomes. So purposes, I don't want to necessarily say these are all purposes. This is a purpose, is to create sperm and egg cells. And just some of the results of the process, the way it happens, you get a reduction division and you get genetic diversity. So when we say reduction division, if you reduce something, you're lessening it. And so it reduces the number of chromosomes. From 2n to n. So if you remember, 2n is diploid. Sorry, I'm getting small squishy handwriting here. And the n is the haploid. So you're halving the amounts of chromosomes in meiosis. So we call it reduction division. And then genetic diversity, we're going to end up with new gene combinations. Which gives us something called genetic variation. So examples again are spermatogenesis. Genesis, if you think about like in the Bible, the book of Genesis talks about the creation. So Genesis, I mean, it's nothing to do with this, but Genesis means creation of. So spermatogenesis is the creation or formation of sperm, and oogenesis is the formation of eggs in females. So just another quick review. For sexual reproduction, you have an egg, which is haploid, so it's N. And with fertilization, again, pictures not to scale, the sperm fertilizes the egg, the sperm is also haploid, and then those together are going to eventually develop into a human, which is 2N. So N plus N is 2N. So reduction division is important because it has to go from diploid to haploid in order to form these gametes so they can fuse again and then be diploid. So let's look here. This is kind of confusing, but these in this, the numbers here are referring to the number of sister chromatids, I believe. So here, we would be 2N. We have single chromatids. And we'll talk about all this in detail in a minute too. But in the S phase, just like in mitosis, the DNA is copied. And so now we have sister chromatids, but they're replicated. And then this first division here, you'll notice there are 2 rounds of meiosis. There's Meiosis 1 and Meiosis 2. So, and after Meiosis 1, the sister chromatids are replicated. I hope you guys can see this. And in Meiosis 2, this is when the sister chromatids that were replicated there will separate, and then you're left with single chromatids. And these are all haploid. So let me look for this paper real quick. Sorry, I'm shuffling here. Alright, so it just so happened that I didn't have access to a color printer the time that I would need it the most because we're using red to show chromosomes from mom and blue to show chromosomes from dad. And so I painstakingly colored all of these in, and I have multiple pages, and it took me a long time. So anyway, sorry if it's a little hard to see, but just comparing mitosis and meiosis real quick. So with mitosis, we start with diploid 2N, and in metaphase, those chromatids line up along the middle. In anaphase, the sister chromatids separate. In telophase, the daughter cells form, and then we have cytokinesis, where they split into two daughter cells, which are still diploid, and they are genetically identical to the parent cell. Over here with meiosis, so we've got Meiosis One, and then Meiosis II is basically the same process as mitosis. So in meiosis, metaphase 1, the homologous chromosomes, and you get one from dad, one from mom. They're going to line up along the middle. And then in anaphase, those homologous chromosomes are going to separate. Like I said, we'll cover this in detail. And then telophase 2, the rest of the cell and cytoplasm and organelles split. And then we have cytokinesis that divides everything else off. And you can see, because of some processes that we're going to talk about, these are not identical. And the processes we'll go over happen during metaphase 1. You'll notice I have some color switching here. I don't know if you can see that. So what that ends up resulting in are cells that are not identical. And by the way, I have a new doc cam. I just need to figure out how to use it, which apparently, as you can see, I don't even know how to show myself right side up. So it could be a process, but hopefully it'll be an improvement. This Elmo here isn't too bad, but yeah, anyway. So on to the actual stages of meiosis. So anytime you hear Meiosis One, I want you to think of homologous chromosomes. Separating. So remember, homologous chromosomes are a pair of chromosomes. You get one from mom, one from dad. And we call them homologous because they're the same size and they have the same genes at the same locations. So we have our same acronym as with mitosis, PMAT. It goes in that same order. But the processes of Meiosis One are a little bit different. So in prophase 1, this is where you have synapsis. That's the homologous chromosomes pairing up with each other and crossing over. And crossing over is one of those processes that results in the cells being unique, and we'll cover that in a minute. Metaphase 1 is when they line up along the middle. And this is where something called independent assortment happens, also resulting in genetic variation. So those homologous pairs line up along the middle of the cell. And what independent assortment means is it doesn't matter if it's not like all the chromosomes from mom are going to be on one side and dad from the other. It's random as to what goes where. During anaphase 1 is when those homologous chromosomes separate. And now that we've separated those homologous chromosomes, with telophase 1, we have those sister chromatids at the poles of the cell. So through the first round of meiosis, we have successfully, hopefully successfully, and we'll talk about what happens when it's not successful, we've separated the homologous chromosomes. So after cytokinesis, after we divide the rest of the cell contents, the cytoplasm, the organelles, we are left with two daughter cells. Since we've only split one time, with 23 sister chromatids. So let's look at that. Again, I have my beautiful colored pictures. So over here, we're showing Meiosis One, what we just covered. So the DNA replicates. So now we have sister chromatids here. In Meiosis One, homologous chromosomes pair during synapsis. So they kind of get together, try to find their pair, their partner. In Metaphase 1, the homologous chromosome pairs align at the center of the cell. And this is where something called independent assortment and crossing over happen. Again, we'll get to that. Anaphase 1, we're going to pull those homologous pairs apart till they are at the poles of the cell. And after that first round of division, we're left with haploid cells that are unique. Okay, let's get to Meiosis 2. So in Meiosis 2, anytime you hear Meiosis 2, I want you to think of, ran out of room here, when the sister chromatids separate. So Meiosis 1, splitting up the homologous pairs. Meiosis 2, splitting up the sister chromatids. And I'm not going to write this all out, but all of these are the same as mitosis. Okay, so in metaphase, they'll line up along the middle. Metaphase 2, they'll line up along the middle of the cell. Anaphase will split the sister chromatids. Telophase, they end up at the poles of each cell. And then cytokinesis, we divide the rest of the cell and its contents. And the results are 4 haploid, just one N, daughter cells. So again, my beautiful drawings. We have the cells from the end of Meiosis 1, and they immediately just go straight into Meiosis 2. So now they're haploid, and cells have one chromosome from each homologous pair. So we split up those homologous pairs in Meiosis 1. Metaphase, those chromosomes align in the middle. Anaphase, the sister chromatids separate and move toward the poles. Telophase, spindle disappears, the nuclei start to form around these new sets of chromosomes, and you end up with 4 genetically different haploid cells, daughter cells. Okay, stay with me. I'm going to make sure this all makes sense by the end, alright? Okay, we don't need to write anything on this one, and this is kind of what I went over. So you've got Meiosis One. This is where synapses and crossing over occur during prophase, and independent assortment happens during metaphase 1. And just a reminder that both of these things result in genetic variation. And genetic variation will become important again when we talk about evolution, because genetic variation or differences among species has to be present in order for natural selection to occur. And again, don't worry too much about that now, but it's important when it comes to evolution. And then again, the two daughter cells from Meiosis One proceed directly to Meiosis Two without DNA replication in between. And then I won't go through these steps again, but you have the steps of Meiosis Two, which are just like mitosis. But we get different results because of that crossing over and independent assortment in Meiosis One. Alright, so meiosis, genetic variation. So what's going to give us genetic variation again are crossing over and independent assortment. I would be familiar with those. I'm going to cover them right now. And again, genetic variation, it's differences in the DNA, which is going to result in differences in genes, which will result in different traits. So I'm going to write that down. So differences in DNA, it's going to lead to differences in genes, which will lead to differences in traits that gives us genetic variation. So crossing over is the exchange of genetic information between homologous chromosomes. So in my black and white picture here, it's not clear to see. So let's look at my lovely coloring again. So this crossing over happens in prophase 1. So the sister chromatids of a chromosome in synapsis, and here I have, again, the red is from mom, the blue is from dad, and as they're kind of getting tangled together, crossing over occurs. So basically they're swapping chunks of their chromosomes. And so the resulting chromatids will look like this. Okay, so this is all still from mom, this is all still from dad, but here on these chromatids, we had some swapping going on. So now it's mixed up between the genes from mom and the genes from dad. So make sure you know that this occurs during prophase 1 of Meiosis One. And it results in new genetic combinations. And this can happen at multiple points along the chromosome. right? So crossing over the homologous pairs for mom and dad kind of get tangled and they just mix up some of their pieces of DNA. And so you can see when we separate the homologous pairs how this is going to cause differences in the resulting daughter cells. The next one that gives us genetic variation is independent assortment. And this is when the homologous pairs align independently at the equator, middle of the cell. So there's no specific order in which they line up. So this occurs during Metaphase 1 of Meiosis One. I'm going to add another bullet point here. It results in new genetic combinations in each gamete. So here's a picture showing just some, actually how many, okay. So we have only 3 homologous pairs here in this example, and this shows all the different possible ways they could line up. So it could randomly be all dad and all mom on one side. Here we have 2 from dad, this last one from mom, 2 from mom, last one from dad. Here we've got a mixture, and just look at all the different combinations that could result. So if we take, with just 3 chromosomes, if we take the way that you figure out how many different combinations you could get, if we take, and this isn't humans because we don't have just 3 pairs of chromosomes, but 2 to the 3rd, so 2 times 2 is 4, times 2 is 8. You can have 8 different possible combinations, which, as you see in this picture, 1, 2, 3, 4, 5, 6, 7, 8. So are you going to get genetic diversity here? Yeah. A whole lot with only 3 chromosomes? Not necessarily. So, how many unique gametes could a human make? So humans have 23 pairs of chromosomes, and 23 to the 2nd power, I'm not going to math it out, but if you do, it's about 8 million. So just from independent assortment, the gametes could have 8 million different possible combinations. So let's write that on this bullet point here. Each human can make over 8 million different chromosome combinations. Which is going to give you over 8 million unique gametes. So, I don't think we really talked about our question of the day at the start. It's hiding down here at the bottom, but it's how come you don't look exactly the same as your siblings? So if you have biological siblings, you notice that you have similarities. Maybe one looks more like mom, maybe one looks more like dad, and you all look kind of like each other, but none of you look exactly the same, with the exception of identical twins, and even then there are probably some differences. So, one human male and one human female together can create over 64 trillion unique offspring. 64 trillion. That's a lot. And that's just counting independent assortment. Oh my goodness. What have I done? Independent assortment. That's not even considering crossing over, where those homologous pairs are going to swap little pieces of their chromosomes. So in short, if you are asked, how come you don't look exactly the same as your siblings? The short answer is because of independent assortment and crossing over during meiosis. So is this a perfect process? No, it's not. And that's where we get errors in meiosis. This is when you get changes in chromosome number. And if you have too few chromosomes or too many chromosomes, it can give you severe abnormalities. And there should be a checkpoint that catches this, and does it always? No, because we have individuals that have these abnormalities, but it should be caught at the M checkpoint. When they line up along the middle and the chromosomes attached to the spindle. So let's talk about some different types of these. So non-disjunction is when chromosomes fail to separate from each other. And actually, 50% of miscarriages in the first trimester are due to changes in chromosome number, meaning the chromosomes didn't make it to the right place. And so that's not going to result in a viable offspring. So a monosomy, if you think of mono, it means one. That's the absence of a chromosome, so you're missing one. So you would have 45. Instead of 46. And tri is 3. So a trisomy is the inheritance of an extra chromosome, meaning you're going to have 3. I was going to say 3 of one pair, but you're not going to have a pair, you're going to have an extra, so there's 3. Inheritance of an extra chromosome, so you're left with 47 chromosomes instead of 46. So autosomal trisomy, it's usually lethal because if you're having these extra chromosomes, you have extra genes, you're making extra proteins, and it can just throw a whole bunch of things off in the body. Exceptions are trisomy 21, which is Down syndrome. Some other exceptions, trisomy 8, 13, and 18 have survived. They're going to have probably some major issues, but they have survived. And for some reason, I don't know why, I kind of want to do some research on this, but trisomy 21 is the most common. And that just could be because they can survive and be born and grow up with this. Whereas with the other ones, they're just miscarried before they can even develop. But this extra protein with the extra 21st chromosome can cause developmental and intellectual disabilities. So trisomy 21 means that these individuals have 3 copies of chromosome 21. There's something they have called the GART gene. And I'm not incredibly familiar with this, but this can cause cognitive defects. And then they also often have heart defects. So here is some of this non-disjunction shown. So here, the pair of homologous chromosomes fail to separate. So one of the daughter cells gets an extra pair and the other gets none. And so as you go through Meiosis II, you end up with extra copies of each of those chromosomes. Gets fertilized by a sperm, and now you have these extra chromosomes here in the what's the word I'm looking for? Zygote? Yeah, zygote. And you're missing chromosomes over here in these ones. So are we going to have problems with these 4 if they develop into a baby? Likely yes. So here, the non-disjunction, that's if the non-disjunction happens during Meiosis 1. If the non-disjunction happens during Meiosis 2, so here you've got your pair of homologous chromosomes. They separate like normal, so we're good so far. This one split the sister chromatids like normal. We're good over here. But over here, for some reason, these 2 sister chromatids didn't separate. So this egg cell has an extra copy, and this is missing a copy. So what we're left with in the zygotes, these 2 are normal, 2N. We're all good. This one has an extra chromosome, so we say 2N plus 1. And this one is missing a chromosome, 2N minus 1. Alright, so keep in mind that this non-disjunction can happen in Meiosis 1 when the homologous pairs are separating, or it can happen in Meiosis 2 when the sister chromatids are separating. So this last part piqued my interest because I feel like I'm kind of an old lady. I just turned 36. And the risk of having a baby with Down syndrome significantly increases after the age of 40 in women. 80% of chromosome number issues occur in oocyte development. So in the woman's gametes and not from the men. And this is because there are pauses during egg development. So I don't know if you know this, but all the eggs that you have, you were born with. So your eggs actually started forming when you were in your mom's womb developing. I think I'm saying that right. And then it pauses in prophase 1 until ovulated. Whereas sperm production never pauses. That's just going all day long. But this is only it's gonna pause during prophase 1, and then when it's ovulated, it will continue the rest of the meiotic process. So, which chromosome is most likely to undergo a trisomy? This is 21. And then here's this graph, but I looked up some facts. So for trisomy 21, at the age of 35, which I have just surpassed, 1 to every 400. I don't know if it's referring to babies or the eggs themselves will have a trisomy 21. But then by the age of 40, it goes way up. So 1 to 105. And if you look at this graph here, this is risk of Down syndrome in live births, a percentage, and this is the maternal age, the age of the mom. This is tiny, I'm sorry, but it's going from 20 to 45. You can see right around here, 38, 39, 40, there's a huge jump. And there are reasons for that I don't physiologically know. I've just heard it as your eggs are getting old. But it all comes down to some of the processes that are used to finish the meiotic division aren't happening properly. That was a very poor explanation. But yeah, so okay, this last page is kind of interesting. So changes in sex chromosome number. So we were looking at autosomal chromosomes and non-disjunction and trisomies. Changes in sex chromosome numbers are non-lethal. So one X chromosome is enough gene dosage to inactivate any others. And so there are these things called bar bodies, which are inactivated X chromosomes. So I don't know if it says this anywhere on here. It does down below, but a normal biological female is going to be XX, and a normal biological male is going to be XY. So, since one X chromosome is enough for the genes that you need, one of these will be activated. And both men and women inactivate extra X chromosomes. So an inactive X is not completely inactivated, and extra X's can have influences on fertility and physical characteristics, and you saw some of these in the pre-lecture assignment. And in females, since you're XX, one of those is going to be inactivated, and it's random which one is inactivated. So, let's talk about kitties. I love cats. And I grew up with a Calico Cat, very similar to this one. So a Calico Cat is the cats that have the orange, white, and black pattern. And Like I said, my cat looked just like this. She was evil, and so we called her Lucifer. I don't know where it came from, but anyway, that was her name. And I'm going to say, we're just going to assume, okay, how do I want to say this? All calico cats, with very, very few exceptions, are female. Because in order for this pattern to emerge, you need two X's. And if they happen to have an X that has a gene for the orange color, and their other X has the gene for the black fur color, then it's random in the different parts of their body, and I don't think it's fully understood why or how, but which one of these are turned on and off. So if this one is inactivated, you're going to end up with black fur in those cells. If this one is inactivated, you're going to end up with orange fur in those cells. So the reason why we say all calico cats are female is the only way that you can have a male calico cat is if they have Klein syndrome and they have two X's. And the chance of that happening, and then for each of these to have a gene for orange fur and black fur, those chances are very slim. So I remember hearing growing up that if you have a male calico cat, it's worth a lot of money. All right. So how many bar bodies would the following produce? So this is something called the Lion hypothesis, and you don't need to know this name. It's just the name of the dude that discovered it. But you can find, let's see, all but one X chromosome in any sex chromosome genotype is inactivated. So to find out the number of bar bodies, you take the number of X chromosomes minus 1. Because you're only going to have one of those that are active. So, if we look at a normal female, XX, how many bar bodies would she have? So we have 2 chromosomes minus 1, so they're going to have 1 bar body. A normal male, XY, they've got 1 chromosome. 1 minus 1 is 0. They need that X chromosome. They don't have any to inactivate. And you guys learned about some of these syndromes in your pre-lecture, so I'm not going to go over those. But poly-X syndrome, poly means many, so triple X here. So we have 3 X chromosomes minus 1. So that would result in 2 bar bodies. And that's pictured above here. And the bar bodies are just kind of the X chromosome, just kind of smushed up against the corner in the nucleus because it's not needed. Whereas the active X chromosome would be out here strung about doing its thing. Klinefelter syndrome is when a male has an extra X chromosome. So 2 X's, 2 minus 1 is 1. Jacob syndrome, XYY, that's a male with an extra Y chromosome. And they only have 1 X, so 1 minus 1 is 0. And then Turner syndrome is missing an X chromosome. And that's going to have 0 bar bodies. Alright, so let's just go back and summarize meiosis really quick. Sorry, I have papers everywhere. Here we go. Alright, so in summary, in Meiosis One, remember the homologous sister chromatids are separated from each other and the resulting cells are haploid. So you're starting here with diploid. Got the homologous chromosomes, one from mom, one from dad. And they find each other and they line up. So this is where independent assortment occurs, is when they line up in the middle. So remember, it doesn't matter if you've got all of mom on one side or all of dad on the other, they line up independently. And this is only showing 2 pairs of chromosomes, so you're not going to see a lot of variation in this sample. But with a human, remember, you're going to have 23 pairs, which gives 8 million different possibilities. So this is where you get some of the genetic variation, and it's not shown here. But then when they, in prophase 1, when they're finding their pairs, they also swap those little parts of the chromosome, which gives us even more genetic diversity. And the results of Meiosis 1, you get 2 haploid daughter cells. You split apart those homologous chromosomes. In Meiosis 2, those sister chromatids, just like in mitosis, are split apart, and the resulting cells are haploid gametes. So we have halved the amount of chromosomes in each of those cells. Now let me find my picture that shows this with, nope, not this one. Hang on, I'm thinking. Oh yeah, that's the wrong one. Okay, this shows with crossing over and independent assortment. So we got DNA replication, the homologous pairs pair up, and it's not clearly shown here with my poor picture, but hopefully yours looks a little better. This is where crossing over occurs. So you're going to have some swapping of the genes between mom and dad on those chromosomes. And then in metaphase, you can see the results of that. So the blue is from dad. You can see the tip of this chromosome is red. This one's blue. They swapped those chunks of DNA. Down here, the tip of this one is blue and this one is red. So they swapped those pieces. And then it doesn't matter which side mom or dad chromosomes line up on. Anaphase 1, those homologous pairs separate. Telophase, we start to reform the nucleus, and then the result you can see after Meiosis 1. We have 2 haploid cells, and they are genetically different because of this crossing over and independent assortment. So straight on to Meiosis 2. Just bringing these cells up. So you have metaphase 2. They line up along the middle there. You're going to separate those sister chromatids. You can still see the differences in the DNA between the 2 cells. And then you end up with 4 gametes that are genetically different, and they're haploid. And if you look closely at these in your book or on your notes in color, you can see the color variations are all slightly different, meaning they're going to be genetically different. Okay, meiosis is rough. It's one of the harder ones to understand. So email me, send me a message on Canvas if you have any questions. And that's it for today. Mav, I've been recording. What's up?
What are those weird lights?
It's my Elmo.
Elmo! Elmo's world.